first of all, that when I met the folks at Synthago and I discovered that literal rocket engineers had decided to turn their attention to making RNA, I was astounded because my first lab job when I was a graduate student was actually making RNA. And I had zero skill in those days. So it was fascinating to see how the sort of the expertise of engineers could be turned to such a seemingly simple task, but in fact they really turned it into a robust technology that is now, as everyone knows, being utilized to make guide RNAs for CRISPR. And as Paul said, to really look to opportunities to spread this technology widely and make it available to everyone that needs access to it. And I couldn't agree more. I think this is one of the real opportunities that we have right now with CRISPR, is that it's a technology that allows anyone with a little bit of skill in molecular biology to manipulate genomes. And that comes with extraordinary opportunity as well as incredible responsibility. And I've really been involved in both of those aspects of the field over the last few years. And so what I wanted to say this morning is really just a couple of things. I wanted to point out, first of all, that for all of you that are using CRISPR as a genome editing tool in the laboratory, you may or may not know that this technology actually came from curiosity-driven research that was conducted by a number of scientists, but including myself and Emmanuel Charpentier. And we've been interested for the last about 12 years in understanding how CRISPR works as a bacterial immune system in bacteria to provide protection against viruses. And it was through studying the molecular mechanisms of this that we understood how a protein called Cas9 works as an RNA-guided DNA cutter, an enzyme that can cut DNA at precise positions defined by the sequence of the guide RNA. And with that capability, then it was possible to harness it as a technology for genome editing in cells that are able to repair DNA breaks using non-homologous end joining or homology-directed repair. And okay, so I just sent my slides, and I'm going to go back to Firefox. Okay, so basically, this has now become a widely utilized tool for manipulating genomes that is based fundamentally on this bacterial immune system. And the way it works, briefly, is that the protein is able to unwind double-stranded DNA at sequences of 20 base pairs that are marked by the guide RNA. And as Paul mentioned, one of the advances that was made by Emmanuel Charpentier and I and our students, Martin Janek, who was a postdoc with me at the time, and Chris Chylinski in Emmanuel's lab, was to combine what in nature are two separate RNA molecules that guide Cas9 into a single guide format. And that really turns this into a robust two-component tool, one protein and one guide RNA, that can be used to define a sequence for cutting in a genome. And by the way, one can do this in a multiplexed format as well, where multiple sequences are manipulated at once. And this has become, as you all know, a very widespread technology now for manipulating genomes, not just to engineer changes to particular sequences, but also to control transcription and to allow imaging of sites in the genome and utilizing basically this fundamental platform for marking and manipulating specific sequences in a genome. Okay, so the slides are up, and I can just start on this one. So this is just to remind everyone that the bacterial adaptive immunity that uses RNA molecules to guide protection in cells is really what CRISPR is all about. It's a mechanism that evolved over eons in microbes to provide an antiviral mechanism. And the thing to appreciate is that in bacteria, which are rapidly growing cells, when DNA experiences a double-stranded break, that triggers rapid destruction of the targeted sequence. So in bacteria, this is a very effective way to destroy viral invaders, and importantly, for the cell to remember that invader and keep a record of it in the genome in the form of this sequence array called the CRISPR that stores snippets of sequence that come from invading viruses. And then if you go to the next slide, so this is a little video that illustrates how we imagine the system works in a natural setting where you have a biofilm of bacterial cells growing. Some of these cells get infected by phage, as in this example here. And if the cell has a CRISPR system, it can acquire a sequence from the phage into this CRISPR array, where each viral sequence is stored between a repeat sequence in the array. So it's a very distinctive sequence. That actually was the first tip-off scientifically that there was something interesting going on here. And then the cell makes an RNA copy of the array, and it looks like the rest of the video might have gotten cut off. But anyway, the RNA copy is what allows the system to work as a programmable system that defines the site of DNA cleavage. So if you click forward, this shows DNA receiving a double-stranded break, for example, with CRISPR. And if these molecules are introduced into eukaryotic cells, where cells have evolved mechanisms of DNA repair, there is and this was established in the field of genome engineering long before CRISPR came along, double-stranded breaks could be repaired by pathways shown here that could trigger disruptions in the DNA sequence or integration of a new sequence of DNA during the process of repair. And that's fundamentally how CRISPR works as a genome editing technology, is to trigger that kind of repair. So if you go to the next slide, this just shows the work that Martin Jinek and Chris Chylinski did originally in our laboratories, where they found that Cas9 is a dual RNA-guided enzyme, so it has a separate CRISPR and tracer RNAs that provide the targeting information for Cas9 in the form of this 20 nucleotide sequence in orange that's on the end of the CRISPR RNA, and with tracer RNA forming a structure that allows assembly of this complex. And then if you go to the next slide, it was research that Martin and Chris were doing that showed that these two RNAs could be combined into a single guide format that allowed a simplified system for programming Cas9 in cells. And this was, for us, that kind of proverbial moment when we realized that a curiosity-driven project to understand bacterial immunity had morphed into something really interesting and different from what we imagined when we started the project, which was an idea for a technology, basically how this could be utilized to trigger double-stranded breaks in eukaryotes or any cell type, and trigger DNA repair, and thereby introduce site-specific genome edits into cells. So on the next slide, I'll just show you, if you go to the next slide, a quick video that illustrates how we imagine this working in eukaryotic cells, where, of course, the DNA is in the nucleus. So by putting a signal onto Cas9 that allows nuclear entry, these RNA-guided proteins can then search the genome in the form of, which is, of course, packaged in chromatin, to find sequences matching the sequence of the guide RNA. And then when that match occurs, the DNA unwinds. There's the formation of an RNA-DNA hybrid inside the Cas9 protein, and then a double-stranded break is created in the DNA. And again, in eukaryotic cells, machinery then arrives to repair the break, and in this example, introduces a small change during the process of DNA repair. And so this system was rapidly adopted after the publication of our work in 2012 by laboratories around the world that began using it to manipulate DNA in cultured human cells, in zebrafish, in plants, and of course in now many other kinds of cells and organisms. So if we go to the next slide, where is this all going? And one of the really exciting applications of CRISPR technology, but it's only one of many, is the opportunity to use it to cure genetic disease. And just a few short years ago, this would have been, which would have sounded like science fiction, but today we're at a point where we can manipulate DNA in cells in the laboratory to correct disease-causing mutations, such as the one shown here that causes sickle cell disease. This is one of the longest known genetic diseases. It's caused by a single mutation in the human beta globin gene that leads to a defective form of hemoglobin that's prone to aggregation and leads to the classic sickle shape of red blood cells in affected patients. Today, we're at a point in the field where clinical trials are already underway for using CRISPR to treat this disease. And there's already the announcement of at least one patient, Victoria Gray, who has had her sickle cell disease effectively cured using CRISPR, which is very exciting for the field, but also brings with it, I think, extraordinary challenges in terms of access and making sure that this technology is available to all of those around the world that need it. And I couldn't agree more with what Paul said at the beginning about the next stage of this field now being thinking about how we make sure that people that need access to this technology can get it. And that's certainly something that I and my colleagues at the Innovative Genomics Institute are working hard to do. So if you go to the next slide, we think that one of the keys to all of this is going to be delivery. So we know that the CRISPR technology works well, but the challenge is how do we get those molecules into the cells or tissues where they're needed? And for all of you that are starting out your scientific career, this is an area where we need real innovation. We need smart people thinking creatively about how to do this and how to do it effectively into specific types of cells. And I feel very confident that this can be done, but we definitely need to have a focused effort to address it. And this will be one way that we can reduce the cost and make these technologies more widely available worldwide. So if you go to the next slide, safety and efficacy, obviously very important. These are two headlines from the past year from commercial and academic clinical trials that are ongoing, and it'll be very important to follow these projects to ensure that CRISPR is safe and that it's effective. And then if you go to the next slide, we of course want to make sure that CRISPR is being used in ethical ways as well. And you may know that there was a recent report released from the National Academies that addresses in particular the use of CRISPR technology in the human germline and putting in place international guidelines for responsible use in that sort of setting, especially for any future clinical use of that kind of application of CRISPR. And so finally, just in the last minute here, I just wanted to say a little bit about the current pandemic that we're in. And one of the things that's so exciting about CRISPR is that it's a very versatile platform. And so in research that was done by two graduate students originally in the lab and then has been obviously now developed by many others, we understood that some CRISPR proteins have the ability to report on detection of an RNA or DNA sequence by cutting a reporter molecule that has fluorescent dyes appended to it. And so this can be done for RNA detection using the family of enzymes known as Cas13, or for DNA detection using the family of enzymes known as Cas12. And we'll be hearing later in the talks today from a team that's working on developing this technology for diagnostics. And of course, there's a number of groups that are doing this both in commercial and academic settings. And I think it's exciting to think about the potential of this to provide a true alternative to PCR or other kind of amplification-based technologies for coronavirus detection in particular to meet the needs of this pandemic, but frankly also gives us because it is a programmable technology, it gives us a tool that can be easily harnessed to detect other viruses and to prepare for future viral challenges that I have no doubt we will have to be dealing with in the future and preparing for. So with that, I just want to if you go to the next slide, yeah, just point out that the platform, this RNA-guided mechanism of recognition, is a very versatile way to manipulate DNA in cells and, of course, in vitro, but also importantly in cells and tissues and whole organisms. So I don't doubt that we'll see continued advance of the technology, exciting, new, clever ways to use that fundamental mechanism to manipulate genomes. We have to meet these challenges of delivery and then controlling the outcomes of genome editing. And of course, doing that in the context of ensuring safety, efficacy, affordability, and widespread access. And that last bullet is really one of the areas where I focus a lot of my attention these days, especially in the context of the Innovative Genomics Institute. And I just want to conclude with a shout out to many people who have contributed to this work. A number of folks in my own laboratory, Audrey, Gavin, Jenny, and Connor in particular, but of course many others that are shown in this picture here that was taken pre-pandemic, pre-social distancing, and then collaborators of ours, David Liu at the Broad Institute, Pete Beal up at UC Davis, Keith Young at Harvard MGH, and of course, all of this started with a wonderful collaboration with Emmanuel Charpentier, who's now at the Max Planck in Berlin. And with that, I'll say thanks, and I think we're going to open it up for questions.